Hi again, biology. This is Mrs. Cozen. We are on our last video for Unit 2. Let's title this Enzymes. Video 3, Enzymes. Okay, I got a joke for you. I know I haven't done a joke in a couple videos here, so here's my latest terrible Laffy Taffy joke for you. What do you call a lazy baby kangaroo? A pouch potato. Haha, that's my new favorite one. I know it's a terrible joke, but just a little something to make you laugh. All right, guys, so let's talk about enzymes. So enzymes are working in your body right now. Every part of your digestive system, except for your stomach, okay, is using enzymes to help break down materials. You guys have enzymes in your saliva, you have enzymes in your intestines, you have enzymes in parts of your stomach, but for the most part, the acid in your stomach breaks down or denatures the enzymes. Okay, so what an enzyme is it's a protein that's going to help you to speed up chemical processes. And one of the ones that I'll talk about a lot is digestion because it's a really easy one for us to talk about. And it's one that you're going to see in your lab that you're going to do about enzymes. So let's talk about first the definition of an enzyme. Okay, an enzyme is a protein. That's not what I wanted. There we go. It is a protein, so of all your 4 macromolecules, okay, enzyme is an example of a protein. And the reason that we talk about this one is that it helps to process all the other macromolecules that we're using, okay? And it helps us to speed up chemical reactions. Okay, catalyst. When you think of the word catalyst, first thing I want you to do is I want you to think about where you've heard it before, okay? Think about where you've heard the word catalyst before, this one right here. Okay, now before I had studied it in biology, I heard it in my literature classes when I was reading about plot twists in a story. And one that I remember we had talked about before I got to biology was Romeo and Juliet. If you guys remember, I can't remember the guy it was exactly, but Romeo's cousin is killed, and it's a catalyst to shift the plot. Okay, so a catalyst is something that's going to change or kind of change the direction of something. In biology, it's a substance that's going to speed up a chemical reaction. Now, a catalyst can be something like stirring up a reaction or adding heat, or it can be an enzyme. So an enzyme is an example of a biological catalyst that helps us to speed up a chemical reaction. And we like that because some of our chemical reactions, if we didn't have enzymes, we'd be in a whole heap of trouble if we couldn't get our chemical reactions to go as fast as they do with enzymes in tow. Activation energy. Activation energy is the energy that you need to start a reaction. Enzymes make it easier for that reaction to happen. So what enzymes do is they lower the activation energy that we need to get a reaction started. So if you think about it like a big hill, if you think about it like, here's my reaction, and here's the energy I need to get it started, and then down the hill, here's my reaction actually happening, okay? My activation energy is this right here. This is what it took for me to get started in my reaction. Now what an enzyme is going to do for you is an enzyme is going to lower. So what an enzyme does is it takes that activation energy and it's going to lower it so you don't have to do as much to get to the end result. Okay, so you see our activation, our hill wasn't as steep, didn't require as much energy to make that happen. So enzymes are awesome. They do a lot to help us out. Okay, lactase, an example of an enzyme. It's one that helps us to break down proteins in dairy products. We use lactase to break down milk, cheese, ice cream. If you've ever heard of someone being lactose intolerant, lactose is the protein that's in many dairy products, and lactase is the enzyme that breaks it down. So if you are lactose intolerant, what that means is that you probably have some trouble with the enzyme lactase working correctly. So when you hear that someone is lactose intolerant or they have a lactose allergy, It might mean that they have difficulty with production of their enzyme lactase. All right, my friend, Benedict's test. The Benedict's test is something that you are going to see in lab, and you'll need to know this for your test. Benedict's test is an indicator, which means it changes color in a substance we're looking for. And it changes color when it hits a monosaccharide. Remember, monosaccharides are monomers. So when Benedict's sees a monosaccharide, it turns orange. All right? And when we take in materials, when we eat sugars and starches, very often they start as polysaccharides. But then we have some awesome enzymes, especially in our saliva, that break polysaccharides down into monosaccharides. So we can do a Benedict's test and actually look at what we're eating and in our saliva, and we can look to see if we have an enzyme that's breaking polysaccharides into monosaccharides. So what you need to know about Benedict's test is that it tests for sugars. It will turn orange in the presence of monosaccharides. All right, now let's take a peek at some of the graphs that you might see that represent enzyme activity. So looking at this, you can see this is the potential energy. So here's my reactants, here's my products. Here's the beginning of my reaction, the end of it. And then here is without the enzyme and with the enzyme. So check that out and see what is happening to the potential energy or the energy that's needed to get this reaction happening when we have the enzyme present. You guys can see, here's my steep hill without my enzyme. The enzyme lowers the activation energy I need, so it helps to actually speed up the reaction. And so I had to put less energy into this reaction to make it happen. All right, think of it like when you want to go down a big hill on your bike or your skateboard. Okay, you have to push up this big hill. You have a lot of energy. You have to push till you get to the top, and then woo, you can ride right down it, or like a roller coaster, something like that. What an enzyme will do for us is it'll help us get to that hill, but then get the same result at the end of it. All right, how about this one? Check these out. Which one of these enzymes is working, or which one of these, I guess it's not which enzyme is working faster, which reaction is happening faster? If you can see, this is the reaction without the enzyme. All this energy is required to get it going, and it ends up with this product. This reaction is happening with the enzyme, only requires this small bit of energy, product at the end. All right, and last one. Enzymes have to have very specific environments in order to work. There are some enzymes that work at low temperatures, high temperatures, certain pH, certain concentrations of salt or potassium. So every enzyme has a very specific job so it can work in a specific place. This graph helps to show that. The question asks which enzyme is working better, or I guess you could say more efficiently at lower temperatures. You can see we have low temperature. So this would be number of molecules. This could be the molecules that are broken down by the enzyme. So this enzyme right here is working better at a lower temperature. Okay. If these could actually both represent the same enzyme possibly. You can see at a higher temperature it's not working so great, but at a low temperature it is working. So this would be an enzyme that you would find in colder environments. And that is the end of video 3.